Hey guys, I'm going to highlight Chapter 4, Section 2. So this is talking about the difference between eukaryotic and prokaryotic cells, and it's also highlighting cell size. So as you can see in this picture, we have a, protic cell and eukaryotic cell. The picture, you can't totally tell by the picture, but if you look at this, the size is a lot different. Prokaryotic cells are a lot smaller. Some similarities between them. Both have a plasma membrane. This is very, very important to know because the plasma membrane is very important for cell function. We're going to get into that in a lot of detail later on. But as you can see, both have them. The prokaryotic cell is surrounded by a cell wall. This eukaryotic cell it's an animal cell, so it does not have a cell wall, but if it was a plant cell, you would see a cell wall there as well. This picture up here is of a plasma membrane. So you can see that there's structure to it, there's function to it. Again, we'll get into that later, but the importance of it is it lets things in and it lets things out. So if we're looking at this picture, this would be the inside of the cell, and this is the outside of the cell. You can see this here. So, I'll come back to that. But anyways, again, both cells have cytoplasm, and that is the liquid jelly-like substance that holds I kind of pointed to the wrong thing, holds the organelles in place. It also has a lot of the molecules and the atoms and the nutrients that the building blocks that the cells need to use for their functions. Both of the cells have DNA, and we talked about this in class. In the prokaryotic cell, there's no nucleus. It is contained in a nucleoid region, which is just an area where that DNA, the chromosomes of the prokaryotic cell, are concentrated. It's not membrane bound. In the eukaryotic cell, the DNA is in the nucleus, which is a membrane-bound organelle. This is really important for you to notice, is that both have ribosomes. Ribosomes are an organelle that manufacture proteins. So when we talk about manufacturing proteins, remember we talked about transcription and translation. That is happening in both of the cells. What question you will need to ask about that is how is it different in a prokaryotic cell compared to a eukaryotic cell without the organelles or without the nucleus. We will get into that later. Both have the ribosomes, so both are making the proteins from the DNA, reading the DNA. Let's see what else. The biggest thing that you should notice is missing from the prokaryotic cell are membrane-bound organelles. So even though there are organelles in a prokaryotic cell, there are no membrane-bound organelles. That is really special for a eukaryotic cell, and why the eukaryotic cells can actually maintain multicellular organisms is because these, if you see them here, you have these membrane-bound organelles. They have a membrane around it. So this membrane, and it's going to be specific for the organelle, but main idea, it is going to separate what is going on inside of the organelle with what is going on outside of the organelle. For example, we know that in a mitochondria, this is the mitochondria helps in producing energy, so cellular respiration. That is able to happen efficiently in a eukaryotic cell because the environment inside of the mitochondria is conducive to that process. In a prokaryotic cell that does not have membrane-bound organelles, they still have metabolic processes going on, but they're not as efficient. They kind of have to use their own cell membrane as it kind of functions as both. One of the really cool things about membrane-bound organelles, which makes the eukaryotic cells more complex and able to be more specialized and sustain multicellular organisms, is that since the inside of the organelle can be different, for example, we learned that enzymes, they denature at certain pHs, and they work better at certain pHs. So the inside of a organelle can have a different pH level than the outside of the organelle, and so that function is going to work a lot better inside of that organelle, and it's not going to affect other parts of the cell. They are compartmentalized. I'm going to write that down. Oh no, I don't know how to spell that. Compartmentalized. Meaning that they are able to have more specific jobs without affecting the other parts of the cell. It is more efficient. And that brings us to the cell size. So prokaryotic cells are a lot smaller than eukaryotic cells because they're not as efficient. So for example, this lysosome right here, it's kind of, it takes waste from the cell. It's kind of like the trash truck if you did the cell analogy poster. So if there's waste inside of the cell, the lysosome will take care of that and it won't affect the rest of the cell and it will move it out of the cell. In a prokaryotic cell without a lysosome, the waste is only able to exit through the membrane. So the cell has to be smaller so that the membrane is closer to all parts of the inside of the cell. That is called the surface to volume ratio. The surface to volume ratio is talking about the amount of surface of the cell compared to how big the cell is, the amount of volume, like the empty space inside of the cell. All cells remain small because the cell membrane is so important for letting certain things in and certain things out. It has to reach all parts of the inside of the cell at a rate that's going to keep the cell functioning properly. If the cell is too big, like this one, you only have a certain amount of surface area to let things in, like nutrients and oxygen. If it was too big, you don't have enough area for the oxygen to pass through, but you have a huge inside that you're trying to keep alive that's not going to get the oxygen it needs fast enough because it's not very close to the cell membrane. That's why when we look at multicellular organisms are big, but their cells aren't bigger. They just have more cells. So as you can see from this graph, as the volume increases, the surface area increases, but at a much lower rate. So we have to keep the cells small so that it matches, the volume matches the surface area so they can get the nutrients they need in and get the waste they need out without affecting the rest of the cell. That's why we see a lot of times cells with specialized shapes. So this is an epithelial cell, so this would be characteristic of like a intestinal lining cell, which we know that the intestines are important for absorbing nutrients. And the reason that these are called microvilli, they have these projections and they're like extended cell membrane. So I know it just looks like a hair here, but it's actually a cell membrane. So things can go be absorbed by these microvilli, and what you're doing is you're increasing the surface area, but you're keeping the volume of the cell the same. So you're able to absorb more with that, with these extensions. Again, to look at this table, because this figure is in your book, the surface area is the outside. The volume is the inside, the amount of space that it takes up. Your surface area to volume, you need to keep it small. You want it to be bigger. I'm sorry, I totally messed up on that. You want it to be bigger. So if your surface area to volume decreases, that is not sustainable for a cell. So if you notice, this surface area right here is a lot smaller. The surface area to volume ratio is a lot smaller than this. This would not be an efficient cell. The amount of surface area is not enough to let enough pass in and the waste come out to keep that cell alive. That is it for this section. Again, this is section 4, chapter 4, section 2.